Think about it. When a living thing grows, what happens to its cells? Does an organism get larger because each cell increases in size or because it produces more of them? In most cases, living things grow by producing more cells. What is there about growth that requires cells to divide and produce more of themselves? Limits to cell size. What are some of the difficulties a cell faces as it increases in size? Nearly all cells can grow by increasing in size, but eventually most cells divide after growing to a certain point. There are two main reasons why cells divide rather than continuing to grow. The larger a cell becomes, the more demands the cell places on its DNA. In addition, a larger cell is less efficient in moving nutrients and waste material across the cell membrane. Information overload. Living cells store critical information in a molecule known as DNA. As a cell grows, that information is used to build the molecule needed for cell growth, but as the cell increases in size, its DNA does not. If a cell were to grow too large, an information crisis would occur. To get a better sense of information overload, compare a cell to a growing town. Suppose a small town has a library with a few thousand books. As more people move in, more people will borrow books. Sometimes people may have to wait to borrow popular books. Similarly, a larger cell would make greater demands on its genetic library. After a while, the DNA would no longer be able to serve the needs of the growing cell, and it might be time to build a new library. Exchanging materials. There is another critical reason why cell size is limited. Food, oxygen, and water enter a cell through its cell membrane. Waste products leave the cell the same way. The rate at which this exchange takes place depends on the surface area of the cell, which is the total area of its cell membrane. The rate at which food and oxygen are used up and waste products are produced depends on the cell's volume. Understanding the relationship between the cell's surface area and its volume is the key to understanding why cells must divide rather than continue to grow. Ratio of surface area to volume. Imagine a cell that is shaped like a cube, like those shown in figure 10.1. The formula for area, length times width, is used to calculate the surface area. The formula for volume, length times width times height, is used to calculate the amount of space inside. By using the ratio of surface area to volume, you can see how the size of the cell's surface area grows compared to its volume. Notice that for a cell with sides that measure 1 centimeter in length, the ratio of the surface area volume is 6:1 or 6:1. Increase the length of the cell's side to 2 centimeters, the ratio becomes 24:8 or 3:1. What if the length triples? The ratio of the surface area to volume becomes 54:27 or 2:1. Notice that the surface area is not increasing as fast as the volume increases. For a growing cell, a decrease in the relative amount of cell membrane available creates serious problems. Traffic problems. To use the town analogy again, suppose the town has 2-lane main street leading to the center of town. As the town grows, more and more traffic clogs the main street. It becomes increasingly difficult to move goods in and out. A cell that continues to grow could experience similar problems. If a cell got too large, it would be more difficult to get sufficient amount of oxygen and nutrients in and waste products out. This is another reason why cells do not continue to grow larger even if the organism does. Division of the cell. Before it becomes too large, a growing cell divides, forming 2 daughter cells. The process by which a cell divides into 2 daughter cells is called cell division. Before cell division occurs, the cell replicates or copies of all of its DNA. This replication of DNA solves the problem of information overload because each daughter cell gets 1 complete copy of genetic information. Cell division also solves the problem of increasing size by reducing cell volume. Cell division results in an increase in the ratio of surface area to volume for each daughter cell. This allows for the efficient exchange of materials within the cell. Cell division and reproduction. How do asexual and sexual reproduction compare? Reproduction, the formation of new individuals, is one of the most important characteristics of living things. For an organism composed of just one cell, cell division can serve as a perfectly good form of reproduction. You don't have to meet someone else, conduct a courtship, or deal with rivals. All you have to do is divide, and presto, there are 2 of you. Asexual reproduction. For many single-celled organisms, such as bacterium in Figure 10.3, cell division is the only form of reproduction. The process can be relatively simple, efficient, and effective, enabling populations to increase in number very quickly. In most cases, the two cells produced by cell division are genetically identical to the cell that had produced them. This kind of reproduction is called asexual reproduction. The production of genetically identical offspring from a single parent is known as asexual reproduction. Asexual reproduction also occurs in many multicellular organisms. The small bud growing off the hydra will eventually break off and become an independent organism. An animal of asexual production is in an animal. Each of the small shoots or plantlets on the tip of a concho leaf may also grow into a new plant. Sexual reproduction. Unlike asexual reproduction, where cells separate to form a new individual, sexual reproduction involves the fusion of two separate parent cells. In sexual reproduction, offspring are produced by the fusion of special reproductive cells formed by each of the two parents. Offspring produced by sexual reproduction inherit some of the genetic information from each parent. Most animals and plants reproduce sexually, and so do some single-celled organisms. You will learn about the form of cell division that produces reproductive cells in Chapter 11. Comparing asexual and sexual reproduction, you can see that each type of reproduction has its advantages and disadvantages when you look at each one as a strategy for survival. Species survive by reproducing. The better suited a species is to its environment, the greater its chance of survival. For single-celled organisms, asexual reproduction is a survival strategy. When conditions are right, the faster they reproduce, the better their chance of survival over the other organisms, including using the same resources. Having offspring that are genetically identical is also an advantage as long as conditions remain favorable. However, a lack of genetic diversity becomes a disadvantage when conditions change in ways that do not fit the characteristics of the organism. Sexual reproduction is a different type of survival strategy. The process of finding a mate and the growth and development of offspring require more time. However, this can be an advantage for species that live in environments where seasonal changes affect weather conditions and food availability. Sexual reproduction also provides genetic diversity. If an environment changes, some offspring may have the right combination of characteristics needed to survive. Some organisms reproduce both sexually and asexually. Yeast, for example, are single-celled eukaryotes that use both strategies. They produce asexually most of the time. However, under certain conditions, they enter asexual phase. The different advantages of each type of reproduction may help to explain why the living world includes organisms that produce sexually, those that produce asexually, and many organisms that do both. 10.2. The process of cell division. What role does cell division play in your life? You know from your own experience that living things grow or increase in size during particular stages of life or even throughout their lifetime. This growth clearly depends on the production of new cells through cell division. But what happens when you are finished growing? Does cell division simply stop? Think about what must happen when your body heals a cut or a broken bone. And finally, think about everyday wear and tear on the cells of your skin, digestive system, and blood. Cell division has a role to play there too. Chromosomes. What is the role of chromosomes in cell division? What do you think would happen if a cell were simply to split in two without any advanced preparation? The results might be disastrous, especially if one of those cells' essential genetic information wound up in one of the daughter cells and not the other. In order to make sure this doesn't happen, cells first make a complete copy of their genetic information before the cell division begins. Even a small cell like the bacterium E. coli has a tremendous amount of genetic information in the form of DNA. In fact, the total length of this bacterium's DNA molecule is 1.6 millimeters, roughly 1,000 times longer than the cell itself. In terms of scale, imagine a 300-meter rope stuffed into a school backpack. Cells can handle such large molecules only by careful packaging. Genetic information is bundled into packages of DNA known as chromosomes. Prokaryotic chromosomes. Prokaryotic lack nuclei and many of the organelles found in eukaryotes. Their DNA molecules are found in the cytoplasm along with most of the other contents of the cell. Most prokaryotes contain a single cellular DNA chromosome that contain all or nearly all of the cell's genetic information. Eukaryotic chromosomes. Eukaryotic cells generally have much more DNA than prokaryotes do. Therefore, contain multiple chromosomes. Fruit flies, for example, have 8 chromosomes per cell. Humans have 46. A carrot cell has 18. The chromosomes in eukaryotic cells form a close association with histones, a type of protein. This complex of chromosome and protein is referred to as chromatin. DNA tightly coils around the histones, and together the DNA and histone molecules form bead-like structures called nucleosomes. Nucleosomes pack together to form thick fibers, which condense even further during cell division. Usually the chromosome shape you see drawn is a duplicated chromosome with supercoiled chromatin, as shown in figure 10.5. Why do cells go to such length to package their DNA into chromosomes? One of the principal reasons is to ensure equal division of DNA when a cell divides. Chromosomes make it possible to separate DNA precisely during cell division. The cell cycle. Cells go through a series of events known as the cell cycle as they grow and divide. During the cell cycle, a cell grows, prepares for division, and divides to form two daughter cells. Each daughter cell then moves into a new cell cycle of activity, growth, and division. The prokaryotic cell cycle. The prokaryotic cell cycle is a regular pattern of growth, DNA replication, and cell division that can take place very rapidly under ideal conditions. Researchers are only just beginning to understand how the cell cycle works in prokaryotes, and relatively little is known about its details. It is known that most prokaryotic cells begin to replicate or copy their DNA chromosomes once they have grown to a certain size. When DNA replication is complete or nearly complete, a cell begins to divide. The process of cell division in prokaryotes is a form of asexual reproduction known as binary fission. Once a chromosome has been replicated, the two DNA molecules attach to different regions of the cell membrane. A network of fibers forms between them, stretching between one side of the cell to the other. The fibers constrict and the cell is pinched inward, dividing the cytoplasm and chromosomes between two newly formed cells. Binary fission results in the production of two genetically identical cells. The eukaryotic cell cycle. In contrast to prokaryotes, much more is known about the eukaryotic cell cycle. As you see in figure 10.7, the eukaryotic cell cycle consists of 4 phases: G1, S, G2, and M. The length of each part of the cell cycle and the length of the entire cell cycle varies depending on the type of cell. At one time, biologists described the life of a cell as one cell division after another, separated by an in-between period of growth called interphase. We now appreciate that a great deal happens in the time between cell divisions. The interphase is divided into 3 parts: G1, S, and G2. G1 phase, cell growth. Cells do most of their growing during the G1 phase. In this phase, cells increase in size and synthesize new proteins and organelles. The G in G1 and G2 stands for gap, but the G1 and G2 phases are actually periods of intense growth and activity. S phase, DNA replication. The G1 phase is followed by the S phase. The S stands for synthesis. During the S phase, new DNA is synthesized when the chromosomes are replicated. The cell at the end of the S phase contains twice as much DNA as it did at the beginning. G2 phase, preparing for cell division. When DNA replication is completed, the cell enters the G2 phase. G2 is usually the shortest of the 3 phases of interphase. During the G2 phase, many of the organelles and molecules required for cell division are produced. When the events of the G2 phase are completed, the cell is ready to enter the M phase and begin the process of cell division. M phase, cell division. The M phase of the cell cycle, which follows interphase, produces 2 daughter cells. The M phase takes its name from the process of mitosis. During the normal cell cycle, interphase can be quite long. In contrast, the process of cell division usually takes place quickly. In eukaryotes, cell division occurs in 2 main stages: The first stage of the process, division of the cell nucleus.